We are very lucky. Dr. Lyle Simmons has joined us from the University of Michigan, just down the road. He's confused. Yeah. And he and I have known each other for 22 years, a long time. Lyle was my RA when we were undergraduates at Elmo. Lyle and I were biochemistry partners. We walked through that, much like some of you have walked through the class. And we went different paths, but got to the same point. Lyle ended up taking a year off in between graduate and undergrad, did some research work as a research tech, and then accepted a position at Michigan State University in biochemistry, working in the academia lab. This is actually a big deal for those of you who aren't aware. Finished up there and said, you know, I'm bored of living in Michigan. Went to a place called MIT. You may have heard of it. Did a postdoc there for a while. It was rather successful. He had job options. He said, you know what? He and his wife wanted to come home. His wife also from Michigan, so they came home. And they joined my old department. So my department did much better. They got rid of me and got Lyle. That was a much better option in their world. Where he has been long enough now, he's now associate professor. He has an extremely active lab, vibrant, young, energetic, go-getters, just like all of you. Funded, yes, just like us. And Lyle also teaches. Lyle teaches about as much as Dr. Newhouse does, same class. So imagine having a functioning lab with funding and teaching intro bio to how many class? 530. They aren't going to fit in here, just in case you're curious. So I'm very grateful for him coming today. He's going to give you a really cool story about mismatch repair, which involves DNA. He's going to be curious. Feel free to ask questions when he's done. He's very, very, very charming and extremely harmless. Trust me, known for a long time. Just give him a hand.
Thank you, Aaron. That was very kind. It really was. So, you know, Aaron was one of the great students at Alma when I was an undergraduate, and we were biochemistry partners, and he carried me through that class as the experimentalist. And you know, Aaron's been a great friend for a long time. You know, Spring Arbor is a fun place for me to come back to. I was here in 1994, competing on a track meet at the indoor facility. Is it still here? There used to be. There was an indoor track. I did win the pole vault, just in case you're curious, in that invitation. But that may have been the last time I won something. So it's nice to come back and have a chance to share my work with all of you. So what I'm going to talk about is mismatch repair, as Aaron indicated, and the model organism that we study is Bacillus subtilis. It's a soil bacterium. You're seeing some images here. One single cell will be outlined in red, and then in green is a green fluorescent protein fusion to one of the replication proteins. Before I get into the science a bit, a couple of things. One is Aaron said you could ask questions of me afterwards for sure. You can also interrupt me during the talk. I'm used to being interrupted. I have 2 children. I'm rarely ever able to get through anything without being interrupted. So please feel free to interrupt me and ask questions. What I don't want are people to be confused during the course of this talk. All right, so real quick, I want to just, before I get into the work a little bit, just give you a sense for our lab. And so right now, this is a little bit of an older picture. It's a tad younger. But I have a postdoctoral researcher, Dr. Lindsay Matthews, and so she did her PhD, and then she came to my lab to continue her training before she looks for an academic position. For PhD students, I have 4. Jeremy, Justin, Brian, and Pete. And so most of the work I'm going to discuss today was done by Jeremy, who is here. I have a master's student, Will Hurst, and then I always have undergraduates in the lab. I love having undergraduates in the lab because of the enthusiasm that they bring to the work. And so 2 undergraduates, Taylor and I, Lane Andrews, who are not here. So let's start at the beginning. I want to make sure that everyone's on the same page. And so let's just, I know you've seen this before, but I just want you to start from this base. So obviously, you know that DNA is the genetic material for all living cells. Okay? And it's, for the most part, a relatively stable molecule. And as there's information in DNA in the form of genes. And those genes are transcribed into a messenger RNA. The messenger RNA is then translated by ribosomes into proteins. Those proteins will then go off and do whatever function they need to do. So you can understand that the blueprint that you have in the DNA is quite quite critical for the cell doing what it needs to do, right? Yes? Good. And so if you mess up that blueprint, if there's something wrong with that blueprint, then all of the downstream steps are going to be a problem, or there's a potential for a lot of problems, right? So just like if you're building something and the plans that you have are wrong, then you're not going to end up with what you want to have. And so my lab and my interests are centered around trying to make sure, you're understanding the proteins and the pathways that make sure that this DNA does the best job to make sure that that DNA is correct. All right, so I said that it's relatively stable, and that is true. But I want you to think about this for a second. Does anyone know how many cells you have in your body?
A bajillion.
Okay, a bajillion, right? Let's go with that. So that's a lot of cells, right? I'm not a mathematician, that's a lot of cells. So during a 24-hour period, every single cell in your body will have to fix 30,000 lesions. And this does not include you going out into the sun and sun tanning, and while you're eating grilled foods, which also create and have carcinogens that can affect your DNA. That's not included here. That's you sitting in a room, just breathing and doing nothing else. Okay, because you have the normal byproducts of cellular metabolism, which are reactive oxygen species, can damage the DNA. And there are about 30,000 lesions that every single cell in your body will need to repair in a 24-hour period. And so if you think about it, for my 50-minute presentation, your cells are going to have to repair roughly 1,000 lesions. I will try not to go over, all right? But whether I go over or not will not affect how many lesions you have in your DNA. All right? For bacteria, we don't measure it in a 24-hour period because they duplicate rather quickly. And so we think about it per round of replication. The bacteria have about 6 lesions that they need to fix per round of replication. And by lesion, I simply mean a base that is modified, something about the DNA that is not natural. It could be a break or a modification of a base. All right, so then in most cases, obviously we're all sitting here, we all seem healthy and everything's going well. That DNA is actually, the DNA damage is repaired, obviously, right? A lot of DNA damage is incurred in your cells, and that DNA, that damage is repaired just fine. If for people that have defects in DNA repair pathways, then they are predisposed to several kinds of cancers, neurological diseases, bone marrow failure, and accelerated aging. And so those DNA repair is very, very critical for your health. And in bacteria, don't think of it that way, but bacteria that have inactivated DNA repair pathways, the cells can die or they can have a reduction in their fitness. And all that means is if you have 2 cells competing for nutrients in an environment, if one has a mutation in repair pathways, it may not be able to compete as efficiently and it will have less fitness. All right. So one thing that my 8-year-old son asks me every now and then, and my wife has asked me, is, well, okay, DNA repair is important for human health. Why would you study bacteria, and why is our livelihood based upon those studies? Okay? Because, I mean, why would you do that? So Bacillus subtilis is a common soil bacterium. It's in the soil everywhere around the world, all over here. It's all over your shoes. It's probably all over the chair, the carpet, likely this area. It's all over your steering wheels. It's everywhere. And it's non-pathogenic, meaning that it's not going to make you ill. It's not going to make you sick. Okay? But what it does have is it has attributes that I love. One is it's somewhat cheap to use. Okay? And then, but that's not the primary driver, right? But you can do just about anything to it you want experimentally. And so if you think about it, if you're in science and you're exploring scientific questions, you can use human cells, you can use a very complicated system, but then the experimental tools that you have at your disposal to answer that question are not the same. Okay? And so if you study bacterium where you have a more simplified system, but then whatever question you're asking, you have genetic tools. You can knock out genes, make fusions, alter genes. You have genomics, it's a small genome. You can do whole genome sequencing. In Aaron's class that I studied, and he talked about not having sequencing. Well, we can sequence a bacterial genome for $150. It's it's very cheap. Now we spend tens of thousands of dollars doing that, but one genome would be $150. We just do thousands of them. Okay. So we have biochemistry. It's very easy to purify many of these proteins and test the enzymatic activity. We have cell biology, which I'm showing here, which is a type of microscopy, a type of cell biology, fluorescence microscopy, where we can label proteins with a fluorescent protein and see where they go and understand how they get recruited to sites. And then we also have the ability to do structural biology, to determine crystal structures of proteins, and then understand the biology. So I love Bacillus because of the experimental tractability of that system, the ease of answering the question with the experimental tools that are available. All right, so our research is focused on mismatch repair. All right, and this is the basic premise of this. Here's your original DNA, and for all of you, you know that A pairs with T and G pairs with C, and that's the normal pairing for the two strands of DNA. And as DNA is replicated, each one of these strands is the template for the new strand that is synthesized. I know this is review for many of you, but let's just get that down. Okay, so if we go ahead and say we replicate this strand here, and so then we're going to have a copy of this. During the replication process, what happens if a T is paired with the G? Right, that's an error, right? The T will not properly pair with G, and you don't want that to exist. You don't want that to stay that. Because if this is not fixed during the next round of replication, this strand serves as a template, this strand serves as a template. And so here, when this serves as a template, you have the same sequence as the original. When this strand serves as a template, now you have a mutation. You have an AT pair, and this is permanent. Right? The system won't know that this is incorrect, because now it's a proper pair once it's been copied. Does that make sense? So here, this is the only stage where the system can catch it and say, okay, that's an error. We need to fix that. If it doesn't get fixed at this stage, and these two strands are replicated, then for one of those chromosomes, this is now a mutation and it's permanent. And so we study this process. And so you can think of mismatch repair as the spell checker for the genome in a way. And then if you think about the errors that are occurring for bacteria, not for your cells, but for bacteria, the replication fork moves at 500 nucleotides per second. It's very, very fast. And that was something that I learned in intro bio as an undergraduate, and that stuck with me. And I was amazed, and I'm still amazed by that. 500 nucleotides. All right, so as we think about mismatch repair, there are two principal proteins that are part of this pathway that are involved in identifying those mispairs, those replication errors, and then catalyzing their correction. The first is mut-S, and you can think of mut-S as the sensor. You can think of S as standing for sensor. And so here's a GT mismatch, and mut-S will bind that. It will find it and bind it. Okay? And then the second protein in the pathway is mut-L. So mut-L, the L we can think of for linker, and what it'll do is it'll then sort of be a matchmaker with the other proteins in the pathway to bring them in and to remove whichever one of these bases is incorrect, and then resynthesize the DNA and hopefully get the proper base. Now, I mentioned that we study this in bacteria, so we got to get L in there. Okay, so now mut-L gets recruited by mut-S. So does mut-S and mut-L, do those exist in your cells? They do. All right. But you have 6 mut-S's. Right. Bacteria have 1 mut-S. You have 4 mut-L's. Bacteria have 1 mut-L. And Aaron can tell you that I would like to study something that is less complicated. It's easier for me to think about. All right. It's easier for me to understand it if it has less pieces. And so we can measure mutation rate. We can knock out these genes. We can knock out the gene that encodes the mut-S protein, or knock out the gene that encodes the mut-L protein, and we can see an increase in error of the replication process. There are a variety of ways to do that. This is one of those methods. You can take a culture of bacteria. These are normal cells. We're just calling these wild type. We haven't inactivated anything. Okay? And then we have a culture of cells where mismatch repair has been activated, so there's no mismatch repair in those cells. And if we take an equal number of cells from these two cultures, we could place them on an indicator medium that has an antibiotic in it. And so to get resistance to that antibiotic, they need to get a mutation in a particular gene. If they get that mutation, they'll be able to grow. If they don't have that mutation, they won't be able to grow, and they will die on the medium. And so this is a very simple way to determine if the overall mutation rate is increased or is the same when you start to knock out genes, and it's a way to understand the effect on the pathway. So you can visualize it in this way. Here we plated the wild type strain. An equal number of cells were plated on both of these plates. 2 colonies, which are circled in black, formed on that plate the wild type strain, which has mismatch repair. For the strain that does not have mismatch repair, you should be able to see that several hundred colonies have formed on that plate. And so although it's scoring for a mutation in a particular gene, overall the mutation rate within that culture has increased, and we can measure it in this way. Now, I'm showing you 2 plates, and that is all fine and dandy. If you were to do this experiment and study a variant protein in this pathway, you might have to do more like that. All right? So it's not as easy as I make it appear. This was one experiment. All right, so I've detailed, every time I see that I think about the money. But anyways, I detail, the plates are pretty cheap. Auger is expensive. I detail the mismatch repair pathway, okay? And we've talked about S, we've talked about L. Those are the 2 major players. And so we've known about this pathway for a long time, for 30 years. Right? It was first found out in bacteria, then people recognized that it was in human cells. It's been extensively studied, in part due to the links to health. And so, you know, whenever you get into anything, you have to think about the questions that drive your interest, okay? And this was the first question that drove my interest. Here's a single E. coli cell. All the DNA has spilled out from that cell. You may have seen this before. It's in a lot of textbooks. For bacillus, one DNA polymerase error, and we know this for sure, will be made every 15.4 rounds of replication. Okay? So the DNA polymerase will make an error that it is unable to correct once every 15.4 rounds of replication. So that tells you how accurate that process is. That still blows me away. That's very, very, very accurate. Right? Such one mutation, or one mispair that needs correction every 59 million base pairs incorporated. All right? So here's the question. You can see all the DNA. Now imagine taking all of this DNA and putting it back where it belongs, which is inside the cell. Okay? Having replication taking place, having transcription taking place, having the DNA bound by hundreds of proteins, about 400 proteins in a bacteria. Now, how is MUDAS? Now, there's about 80 MUDAS dimers per cell. How can MUDAS identify a single mispair when it needs to act, which is in about 15.4 rounds of replication? How does it do it when it has to contend with all of these issues? And that was a question when I was in my postdoc that drove me. Does anyone have any thoughts? Any of the students? Go ahead.
Does it bind and slide and look for aberrant gallons?
In ways, yes. It certainly is a sliding. If you take MUDAS and you have a mismatch repair, it does slide, and then when it binds the mismatch, then it will actually stay on the DNA. And for many years, the model has been it must scan the whole genome. Now, does that seem feasible? 80 copies and 4 million bases. To me, it didn't seem feasible. People argue it is. To me, I couldn't wrap my mind around it. Now, just for the numbers, just to show you the importance of mismatch repair, strains that have mismatch repairs for the cells, they'll get one mutation, will escape both the DNA polymerase proofreading and mismatch repair once every 940 rounds of replication, so about once every 3.6 million bases. So you can see how mismatch repair really increases the accuracy. All right, so the first question that we're going to address and we may only get through that question. That's okay. Is how does MUTS find that mismatch? Okay. The second question that is still unresolved, and this has been unresolved since it was known that eukaryotes have mismatch repair, is the following issue. If you identify, and when mismatch repair has to do two important things. It has to find this mismatch, okay? But then there's a problem. Both bases are normal, right? They're just mispaired. Neither is, in the case of a damaged base, the recognition, well, the proteins that bind to the damaged base, they know which one is wrong. One is broken, right? One has a chemistry about it that indicates it's wrong, and the protein sees that as, well, this is broken, and then it fixes it. But here, the G is correct. The T is correct. They're just mispaired. It's the wrong pairing. So, which one of these is the error? And the system had to figure that out. Now, E. coli, it's a clever little bug, if we can call a bacterium clever. But, and it has a neat mechanism of doing this. However, very few bacteria do this, and no eukaryotic systems do this. And what E. coli does is it methylates its DNA, and that methylation is coupled to this process. Now, other organisms methylate their DNA, but in this case, actually many do. In this case, methylation is also coupled to mismatch repair. So what it does is that the DNA that's been in the cell for a while, so the template strand will have methyl groups on it. The newly replicated strand is unmethylated, right? And so then when MUT-S binds that mismatch and recruits MUT-L, MUT-L recruits another player that will cut the DNA that is unmethylated. And since this strand is unmethylated, the system therefore assumes this is the new strand and will repair that. Okay. It's a beautiful, beautiful system. However, for basically, I don't want to say all other bacteria, but almost all other bacteria, and all eukaryotes do not utilize that system. They methylate their DNA, but it's not strand specific. Because sometimes the template strand doesn't have it, depending on where it is. And sometimes the new strand will have it, not immediately, but it will have it. And so it's not strand specific. So it can't utilize that system. They don't have this protein which cuts that DNA. They don't have the specific methylase. And so then we come to this. Well, how does the system then, if you're presented with this kind of situation, MUT-S binds this, it recruits MUT-L, how does it know which one is wrong? And that is still an outstanding question in the field. We are closer to answering that question, and we have now a better way, I think, of going about doing it. But this is something that scientists have thought about for frankly 20 to 30 years, and we don't have a great answer. And that's something that my lab is still investigating. All right. So what I want to know, what I want to understand is how mismatch recognition and report are coordinated in live cells. How does MUT-S find that mismatch, that needle in the haystack? Okay? And then I also want to know how the pathway recognizes an error-containing strain. And so I will answer the first question for you, and I will give you a hint as to one possibility for the second question. And so what mismatch repair does is it searches the new DNA as it's being replicated. And it comes in and it searches the DNA just after it's synthesized. And so it targets its search. So if I were to test you, this is what you would want to know. Right? It's a good thing you're not getting tested, but this is really what you have to come away with from this talk. It targets its search to new DNA, and it recognizes the mismatches at that site. It's very difficult for it to find mismatches in other places. It can do it, but it's hard. And it does that by interacting with components of the DNA polymerase. And so the DNA polymerase is the enzyme that's making that DNA, and MUT-S will bind to it and then search that new DNA that's being synthesized at replication. The other one, which you don't want to think about this for standardized exams, frankly. So just think about it in this room, but because it's not going to make sense for like an MCAT kind of answer. But how do you identify the error-containing strand? And what we are finding and what we're really investigating now is this distinct possibility, which is we've learned over the last few years that the DNA polymerase, instead of using a deoxyribonucleotide, it makes what we call a sugar error, and it puts in a ribonucleotide. And so ribonucleotides make up RNA, right? And RNA polymerase uses those to make RNA, right? And the DNA polymerase is very good, in theory, at keeping the RNA out of the DNA. We talked about the stability of those molecules. RNA is not stable. DNA is stable. You don't want the RNA in the DNA. For those of you that have taken intro bio, you know that RNA primes DNA replication and that RNA is removed. And that was when it was really only thought RNA was there, okay? But that is not true. The rate of single ribonucleotide incorporation into genomic DNA is actually quite high. It's about once every 2,300 base pairs. So for a DNA mismatch, it's once every, 59 million. For an RNA, putting RNA in there, it's once every 2,300 base pairs. And so it's a very, and I can describe that in greater detail, but the bottom line, the reason this was missed is the DNA polymerase active site has mechanisms to keep RNA out, but if ribonucleotides out. But if you provide the DNA polymerase with the in vivo concentrations of those nucleotides, ribonucleotides are much, much higher than deoxys. Like for some of them, it's 600-fold higher. Okay? And so when you give it that kind of concentration, then the polymerase's ability to exclude those ribonucleotides starts to, still does a pretty good job, right? 1 every 2,300 base pairs, but you're replicating a 4 million base pair genome, then you can see you're going to need a lot of those. All right, so it basically, if a ribonucleotide goes in and that gets taken out, that would be in the new strand, and that may very well provide at least a partial signal for that strand. So we're going to do some bacterial cell biology. I just want to, it's very simple. It's a very simple system. You have the membrane that's stained in red here. We have this fluorescent membrane dye that will stain the cell membrane. And then the DNA is stained with something called DAPI, that's in blue. One thing that you notice is that the chromosome here takes up basically the whole cell. We're about to get cell division here. This is just after DNA replication, and we're about to have the septum close. It's getting ready. And then we'll have one chromosomal copy in each cell. Here's one where it had divided, so you have the cell here and a copy of the chromosome. And so what I wanted to do initially was say, okay, we understand mismatch repair. We know a lot about biochemistry. Let's go inside of the cell and sort of try to understand how it functions. And so this is the experimental setup. We're going to take mut-S protein, which senses the mismatch. And then we're going to fuse it to green fluorescent protein, which is from a jellyfish. Okay? Sound cool? And the green fluorescent protein is going to fluoresce, as it suggests, and then you can see where that protein is inside of a live cell. So we're going to do that with mut-S and mut-L. And then what we can do is we can just think of this as a base analog, 2-aminopurine. I will abbreviate it 2AP. We can just grow cells in that. The DNA polymerase will put that into the DNA accident. It will increase the error rate of the DNA polymerase. So again, 1 every 15.4 rounds of replication. I mean, it's kind of hard to see. You can see it, but it's going to be tough if you don't do something to increase that error rate. All right? And then what we do, and we can simply ask, do we see mut-S and mut-L, do they organize in a way inside the cell that we can interpret? Or are they diffuse all over the place, which would suggest more of a scanning the whole chromosome type mechanism? So if you do that, you get this result. So in untreated cells, you're seeing a group of cells here. Again, the cell membrane is in red. The mut-S GFP is in green. It is diffuse in these cells. But you will see what we call a focus, which is a concentration of protein that forms an assembly that we can view. And so hopefully you can see these green dots. Can you see them? And so that's mut-S and mut-L, and they're responding to that 2-aminopurine treatment. So we go from foci in 7 to 10% of cells to foci in 40 to 50% of cells if we treat with this base analog that would be incorporated. And so if you analyze this, which we did, the position of these foci suggests that they are at the site of DNA replication. Okay? But one thing that I could never really wrap my mind around is, well, if MUDS is always at the, is it always at the DNA site of DNA replication, or is there something else that causes it to go there? Because if it's always there, then shouldn't I always see it in untreated cells? Shouldn't it be at the site of replication all the time? And I used to make all these fun arguments about why we don't see that. And I turned out I was correct. Okay, but if you don't have the data, then nobody. So how are we going to do this? Okay, how are we going to do this? So you need to see for these foci, we've quantified how many molecules are in that, and in those foci, and it's about 8 dimers. So you need about 16 different MUDSs to really see this thing. And so what I want to do, which seems kind of like a dream type scenario, I want to see 1 MUDS in the cell. Yeah, so I saw some eyebrows go up. And that sounds good, doesn't it? That's enticing. And so there are about 4 people in the world that can do this. I'm not one of those people. But one of the people that can, she's in chemistry at the University of Michigan. And so we began to collaborate with her. Her name is Julie Bettine, and she's a biophysicist and a chemist, and she can image single molecules in live cells. She hadn't done it with this type of system yet. And so what we're going to do is I'm going to show you single molecule analysis of proteins. Okay? So this is in my view, this is pretty cutting edge. And so what we're going to do 2 things. The first setup we're going to do is we're going to take a component of the DNA polymerase and we're going to translationally fuse a fluorophore to it, so a protein that will fluoresce. And what we're going to do is we're going to express 1 or 2 copies of this per cell, and then we'll be able to see that single DNA polymerase molecule. We're going to leave the rest of the, there will be quite a bit of this that will be unlabeled that will allow for replication to work. And so if you think about a replication fork, what we should see, it's kind of light here, there is a strand here. Basically, you have your leading, the light green isn't shown, but anyway, you have your leading and you have your lagging strand. We won't get that level of resolution, but what we will be able to see is the DNA polymerase, where it is in the cell as replication is going on. And so if we first analyze the DNA polymerase, this is a raw image where you can see the outline of the cell, and you're seeing a single molecule that is fluorescent. And then the DNA polymerase in bacillus will occupy somewhat distinct subcellular locations. And so sometimes it's at mid-cell. If it's at the quarter position, it means there's another one here, but we're imaging a single molecule, so we don't see that other position. And so then what we can do is we can take a bunch of molecules that we have imaged, like 30 or 40 or 50, and we can take their position, and then we can stack them all and see where they are into one image. And by we, I mean the people that work them out. And what you find is all of those molecules were in this very discrete location over the cell cycle of the cells. And so the DNA polymerase, at least this component of it, is quite confined. And so there's a little bit of tension in the field. Does it really move like a train on a track, or is it more of a factory where the DNA is pulled in, replicated, and pushed away? But in bacillus, it's more, at least under these growth conditions, it's more of a stationary, not stationary, it's more of a confined complex versus riding sort of along on the template strand. It's a little bit more static. It will move, but only within this confined region, and then basically the DNA is pulled in, replicated, and pushed out.
Is it associated with memory?
That's not clear, okay? And
Do you think it is?
Not really. Okay. And the reason is because we often will see it dead center and not off to the side. when you image a lot of cells. You do occasionally see off the side. There are many, many reports throughout the literature where DNA replications proteins have some kind of membrane association. And so I cannot say that it doesn't do that, but I'm not sure how functional that is. Right? It's hard to, if someone shows that, that's fine, but then you need to show that that's important. Right? And so for this DNA polymerase complex at the quarter position, this one was also quite localized. So now that we have a location information for the DNA polymerase, we can now interrogate MutS. So this is the setup for MutS. MutS and MutL are in an operon. And I'm sure you guys have learned about operon structure. And so what we want to do is take this PAM cherry thing, this red thing, and the PA stands for photoactivator. And so it will not be fluorescing normally, but if you hit it with a laser, then it will fluoresce. And it'll turn on, you can see it. And so there are two concerns here. The first is we need to have MutS fused to this fluorophore, this protein fluorophore, and we also need MutL to be expressed. Okay? So we did a little bit of genetics here, and so MutS is fused to PAM cherry, and then we have a new ribosome binding site, so MutL can be translated and expressed downstream. Okay? And so then the first question before you do any imaging would be, does that thing function? Because if we fused, if you think about it, if you take a protein and you fuse another protein to it to see it, right? It's kind of like you guys throwing on a 50-pound backpack, perhaps, and walking around, right? It's going to interfere with your daily activities. And so when we put this protein on MutS, then the question becomes, well, is it able to do mismatch repair? If it's not, then the imaging is irrelevant, right? If that protein can't function, then the imaging doesn't matter. So we test that. Oops, jumped ahead too far. Okay, so we did that, and so here's mutation rate. So if we knock out MutS and MutL, and if we do that plate acid that I showed you and count those colonies and generate a mutation rate, the mutation rate for the strain lacking MutS and MutL is quite high. If we do that for wild type, it's low. If we do it for the strain with MutS fused to PAM cherry, it's also low. So it's 97% accurate. Does that seem reasonable to you guys to then move forward and do the microscopy? All right, sweet. So what we're going to do next then is all of the MutS is labeled. Okay? All of the MutS within this cell has that label, but we want to see one. And I told you that it was photoactivatable. Okay? So if you have the cell and you shoot it with a laser, it's going to activate one. This molecule will then move in the cell. We can track its movement, speed, and location. And then we can repeat that on that same cell and activate another MutS. Have it move around, do the exact same thing, do it 30 times. Okay? And then move on to the next cell and do it again. And then move on to the next cell and do it again. And track all of those and do it under different conditions, under different genetic circumstances. And so this is what we find. Now these cells have not been treated with 2AP, which was the condition that caused those large focal assemblies to form. And this is untreated, where we don't really see an assembly using conventional fluorescence microscopy. Okay? Now we're doing the single molecule imaging. So here's the outline of the cell. This line is a division where I'm showing what's called the palm reconstruction. All that means is that is the heat map of location. If it's more white, it means there are more MutS molecules there. If it's black, there were none there. Okay. And then what you're seeing on this split screen is the individual traces that a single molecule would have taken. Okay. And so if you analyze this data, what you would find is that a lot of MutS molecules in cells that are untreated in the error rate of replication process is quite low, are at one particular location. They move throughout the whole cell, but they either converge on this location or originate from this location and leave, and sometimes come back. And so then if we label the DNA polymerase, again, I mentioned to you that it shows a very confined and distinct localization in the cell. So with these two fluorophores, these are different colors, so we can do it in the same cell and see where they are at the same time by this single molecule analysis. All right, so that's what you're seeing here. So the blue here was the DNA polymerase. The red here is MUDAS. And what you should see, MUDAS tracks, I mean, this moves away from that site. A lot of the tracks overlay or overlap where the DNA polymerase is located. So you can analyze this in a different way, and that would be, well, what is the speed of the molecule out here, here, and out here? And so we can take the speed of movement and then group it based upon how quickly it's moving. And so what we find, and we're indicating that by dots here, for slow-moving MUDAS molecules, they're at this white, colocalized with this white, most of them. For medium-moving MUDAS molecules, most of those also colocalize with that white, which is the DNA polymerase. And then most of the fast-moving molecules, which are blue, are away from that site. Okay? So the bottom line here is that when MUDAS is at the site of the DNA polymerase, it moves much more slowly than it does when it is away. And I can quantify that for you in a different way by looking at one particular trajectory. And so what we see here is we have where we took a trace of a DNA of a MUDAS molecule, okay? And then this is where it's hitting the area of the DNA polymerase, that confined location. You can see that there's quite a bit of squiggling going on. So here is your separation distance from the replicome. So this is where the replicome is located. And here's MUDAS moving, and now it comes into that region. Okay, this is the region of confinement of the replicome, the DNA polymerase. And so now we can track the speed. So here's this, the speed is increasing. Okay, so this is, the speed is quite random. The speed of the DNA polymerase, of the diffusion of it, is this dotted line. Once MUDAS gets to that region, it slows down and it matches the diffusion rate of the DNA polymerase. So once it's there, it matches the DNA polymerase, and then when it leaves, it begins to do its, it speeds up quite a bit, and it has more of a random pattern. All right, so we've learned a lot by doing the single molecule analysis. And so now what we can do, we characterize that, and this is more of an observational, it's like we're watching it, we're seeing what it's doing, we're treating, we're scoring, we're looking at speed and so on. What we can do now is begin to make some mutations, so take some MUDAS variants that have different properties and then ask what happens. And so I'm going to display that data in a little bit of a different way. And so here we're just looking at the heat map. And by heat map, the positional, how often a position is occupied by MUDAS. The hotter it is, the more MUDAS molecules spend their time there. And we take their position, and then it's sort of like that stacking where we're taking them and asking where they are. And so for the DNA polymerase, I told you that it's confined. And so here it's green is where the DNA polymerase is. And the blue, the cold here is where we don't see it. And so then for MUDAS, we see, although it's a broader distribution than what the DNA polymerase shows, and we expected that because it does track throughout the cell. But it still is co-localized to the site where the DNA polymerase is located. Right? This is in cells that are untreated, so the error of replication is low. If we treat cells with 2-aminopurine and increase that error rate, these are basically unchanged. And so what I've learned, or what I learned from this, is that MUDAS is always present at that site, and it is always, should be, always interrogating that unit and trying to identify errors that could be. So we can test them. So what we're going to do next is we're going to take a MUDAS molecule, a MUDAS protein, that cannot interact with the DNA polymerase. And this one is called MUDAS 800. So we know from biochemistry that this particular mutant is unable to bind one component of the DNA polymerase. And then what we're going to do next is we're going to take a MUDAS mutant, or variant protein that is unable to recognize mismatched DNA. Does that make sense? So we can make an amino acid substitution in MUDAS that will prevent it from being able to determine mismatch-containing DNA from complementary DNA. So, in this case, the mutant of mutas that is unable to bind a mismatch, what would you expect to happen to the mismatch repair pathway? Would it function? No, it's completely inactive, right? Because its ability to recognize that mismatched DNA is gone. And it's a single protein that can do it. For the one that cannot interact with the DNA polymerase, what do you think happens as far as the function of the pathway? Just think about it for a second. It's 90% defective in the mismatch repair. Right? It's not a complete defect, but it's pretty defective. So what we want to know then is where are these proteins? Okay. So we'll start with mutas. If you call 800, it's missing a C-terminal part of the protein that's important for interacting with the DNA. So here's your heat map here of mutas 800. Okay. Since it's not, the colors around the DNA polymerase are not very hot anymore, right? Not very bright. It means that it is really very diffuse throughout the cell. So it has lost the ability to be recruited to where the DNA polymerase is. Okay? This is in cells that are untreated. If we treat with 2-aminopurine, there is very little change in that distribution. Okay? Now, for mutas F30A, which is this variant of mutas that we engineered, which cannot interact with it, it cannot recognize a mismatch. I do want to show you the biochemistry really quick so that you believe me. And so we purified mutas. This is an SDS pH gel. This is a molecular weight marker. So there are no other bands on this gel, indicating that the mutas is pure. When we purified mutas F30A, there are no other bands on this gel. So what we do is we take a DNA substrate with a mismatch, and we ask if mutas can bind it. And so here's regular mutas, which can bind a mismatch. And so as you increase mutas concentration, you see that it binds to this DNA, okay, showing that it binds mismatched DNA. If you give mutas the regular protein, the normal protein, DNA that is complementary, meaning no mismatch, that's blue here, it does not bind it. If we take mutas F30A and give it mismatched DNA, it does not bind it. So, before I show you the localization, I only have a few minutes here. What do you think happens with mutas F30A? I just want you to think about it for a minute, right? So, this cannot bind a mismatch, but it has the region of the protein that does interact with the DNA polymerase. So what do you think its localization looks like? There are no wrong answers. Well, there are, but it's not. It's okay, right? Because we make in science we make a prediction, and sometimes I'm right, and quite frankly, a lot. I shouldn't say a lot, but I am wrong. I mean, I make a prediction, and sometimes I'm right, and sometimes I'm wrong. I mean, that's just the way. That's why you test your prediction. So just you don't have to say the prediction. Just think about it in your mind. What do you expect? Mutas F30A can bind the DNA polymerase. It cannot recognize mismatched DNA. Right? So here's mutas F30A, and here's the wild type protein that can bind a mismatch. What you see is that in untreated cells, its distribution is basically the same as what we see for wild type. If we add 2-aminopurine, the base analog that will form a mismatch, that is unchanged, similar to wild type. So mutas F30A, which cannot recognize a mismatched DNA, is still recruited to the site of replication to interrogate the DNA there. It just can't do. All right. So what we've learned so far is that many of the ribosome components are confined. So that might be a surprise to some of you. But for some bacteria, for some organisms, the DNA polymerase is more of a complex, and it can kind of be in one area and replicate the DNA. We've also learned that MUDAS moves quite a bit around the cell, but it moves to that site. And then we also learned that MUDAS movement is slowed at the replisome, and by replisome I just mean DNA polymerase complex. Okay? And it's being recruited there even if it's unable to biochemically to bind the mismatched DNA. I have a few minutes, so I want to take you through the extension of this project. One thing I have done here is this is all unpublished data. I wanted to share that with all of you. And so we've seen the cell biology, right? So far you've seen the single molecule image. Is there a way to test whether or not it's actually on the DNA and on the same DNA that the DNA polymerase is on in the cells? So I want you guys to think about that for a minute, or less than a minute. Is there any type of experiment that starts inside of a cell where you can determine if a protein is bound to a particular region of the DNA? And so this is actually the one where all my money goes. There's a method called Chromatin Immunoprecipitation followed by sequencing. Shift C is how that's abbreviated. So what you can do, where we have this co-localization of MUDAS and the DNA polymerase, and so I want to know, is it actually on the DNA? Okay. So what I'm going to do is, so chromatin just means DNA and protein, okay? And so if you have, in normal cells, if you have DNA bound to protein, you can treat with a cross-linker that will covalently link the protein to the DNA. Okay? Then you burst open those cells. You take an antibody that's specific for MUDAS, or your protein of interest. It will bind to that protein. You then rip it out, and the DNA comes along with it. Okay? You then release that DNA, and then sequence it. Right? Once you sequence that DNA, and you have that information, you can go back to the genome sequence and figure out where it was. It seems very complex. It's not. It's just a little bit. But it's quite straightforward. So this is what we're going to do. We're going to take cells. I'm not going to show you the control in this case, but we took cells that were not replicated, and we did this experiment. Okay? And the answer is MUDAS shows no enrichment anywhere on the chromosome. Enrichment means it's occupied there quite frequently. Then what we're going to do is we took cells and we synchronized them so they would all initiate replication at the same time. And then 10 minutes in, we block it. So for the cells, it's a circular chromosome. You have a single origin of replication. Once you have replication initiation, forks move bidirectionally around that circle and terminate at the terminus. And so we're going to stop it here. We're not going to treat with 2AP so the error rate is low. We're going to then cross-link the protein to the DNA and figure out where these proteins are. So if you do that, you get this result. And so here is this is fold enrichment, and this would be chromosomal position. You can think of it as taking the origin region, which is located here, and the terminus, and kind of, in a sense, cutting that DNA and laying it out flat. So the terminus is here and here. So this region right here is represented by this line and that line. And the origin is represented, this origin is represented in the middle. And so early on in the replication process, 10 minutes in, we see that MUDAS is highly enriched in that region. One of the DNA polymerases, called polymerase C, is highly enriched at that region, and the other DNA polymerase, DNA E, is also highly enriched in that region. So it shows that they're all bound to the same DNA segments, the same general area of the chromosome at the same time in cells that are not getting replication errors. All right. So what I want to do real quick here, out of time, I believe, right? I want to very quickly about MCDB. If you're interested in doing a PhD, then I recommend you look into MCDB at Michigan, where your wonderful professor was trained. He educates you very well. He was trained there, so you can get the same level of education. In this department, we have people that do biochemistry, animal physiology, neurobiology. You know, I'm more of a microbiologist. Cell biology, plant biology, and then developmental biology systems. One thing about a PhD that you may or may not know is you actually get paid to do it. Did you guys know that? This is gonna upset all the professors, but you get, I think it's about $29,000 a year. Now, what I know is my total cost is $57,000 per year for a student. Okay, due to their benefits and everything, and you know, medical insurance and stuff like that. So what you do is you would come in and you would work in lab and generate a thesis research that is, you know, develops a new topic or enlightens a new topic, and it takes about 4 to 6 years. You don't get your summers off, but you also get paid, and it pays for your education. And you would get a stipend of about, I think it's about $29,000 a year. And so it's about, you know, on average, they say 4 to 6 years. Most people graduate about 5 and a half. And then you can go down a path, become a faculty member. You can go work for a company. You can actually go into science law. You can do a variety of careers, some of which are very lucrative, some of them. But you should do what you want to do. So it's a great option. Take a look at the website. Thank you so much for your